Good morning, my name is Ashton Ryan. I'm a 15-year-old sophomore at Rochester High School. The title of my project is Zooplankton Studies in Lentic Ecosystems, Phase 4, Regional Variations. I would like to thank the Science Department at Lakeland Community College for the use of various equipment and materials as well as a lab space. I would also like to thank my sponsor, Mr. Lyndall Robinson, a retired biology professor, and Ms. Godar, my biology teacher at my high school, for their assistance and guidance throughout the project. The purpose of my project was to determine if various zooplankton populations in Lake Springfield exhibited any regional fluctuations. My hypothesis was that the various zooplankton populations in Lake Springfield should experience certain regional variations. This project is a fourth-year study of zooplankton populations in Lake Springfield. 3 years ago, my initial study was a qualitative analysis where I basically just determined what types of zooplankton were out there. Then my project after that was a seasonal fluctuation study where I just determined that the zooplankton populations exhibited seasonal fluctuations, and last year's project proved the existence of diurnal fluctuations of zooplankton populations in Lake Springfield. This year's project focuses mainly on the existence of regional variations of zooplankton populations in Lake Springfield. The overall significance or importance of this project is based on the fact that zooplankton are good indicators of water health and ecosystem health due to the fact that they are susceptible to many changes in the environment and that they are a vital link in the food chain. This makes them a good indicator of water health and water ecosystem health. Luminology is a scientific study of lakes. Lakes are large inland lentic ecosystems with many physical, chemical, and biological parameters, most of which these parameters can fluctuate daily, weekly, monthly, or yearly. The lake involved in my project is Lake Springfield, which is a 4,600 acre reservoir located south of Springfield. The temperature of a lake is the measure of the amount of heat energy present. The temperature of water doesn't change as rapidly as that of air because water has a higher specific heat or heat capacity compared to that of air. The amount of impinging light hitting a lake surface can also impact its temperature. 1% light level in a lake represents the level at which only 1% of the total light hitting the lake's surface remains. Since there is no light below this level, no photosynthetic activity will be occurring, which means that most zooplankton populations will not migrate below this level. Carbon dioxide, dissolved oxygen, and pH are all chemical parameters that can fluctuate as a result of many factors. The most important of these factors being the metabolic reactions of photosynthesis and cellular respiration occurring within many aquatic organisms in the lake. High levels of both phosphates and nitrates can also impact zooplankton populations. An increase in both of these chemical factors can increase the phytoplankton populations, which zooplankton feed on. An increase of these nutrients can be attributed to agricultural runoff, sewage discharge, and burning plants and purification plants. High levels of both phosphates and nitrates can impact a process called eutrophication, which is basically the natural aging of lakes. And it goes as lakes go from a poor feeding or oligotrophic state to a true feeding or eulotrophic state. The increase of these nutrients can cause an unnatural acceleration of this process, which can eventually lead to the untimely demise of a lake. The three major groups of zooplankton found in most lakes are rotifers, cladocera, and copepods. Rotifers are in the phylum Rotifera, while both cladocera and copepods are in the phylum Arthropoda. Copepods can be observed as either adults or juveniles called nauplii. All zooplankton are motile but small enough to float in upper layers of lakes. Rotifers move around by anterior wheels of cilia, while both cladocera and copepod use various antennae to move around. Zooplankton species can be classified as either herbivores or carnivores depending on their food source. Regional variations in zooplankton populations refers to the change in body size, abundance, and composition of zooplankton populations in a lake. The regions of the lake can have variations being caused by both physical and chemical fluctuations. 3 test sites were selected for this project. The northernmost test site being located north of Lake Springfield near Sprawling Dam and CWOP, while the central test site was located near Lindsay Bridge Boat Launch, and the southern test site being located at Bridgeview Park near Chatham. All 3 test sites were visited on 5 separate days during a 2-week period in June. This was done so that the meteorological conditions would be similar and minimal fluctuations would occur. To further decrease fluctuations, all the collections were taken within a 2-hour time frame. During each of the 5 collections, the following physical procedures were conducted. The site depth and 1% light level was measured using a Secchi disc. Both air temperature and water temperature was measured using a Celsius thermometer, as well as the impinging light being measured by using a handheld portable light meter. A water sample was then collected and transferred into a 1,000 milliliter beaker to be used for chemical testing in 5 different chemical parameters. The 3 of these chemical parameters include the carbon dioxide, dissolved oxygen, and pH levels, which were all measured on site using the appropriate materials and instructions in a hot water ecology test kit. All the phosphate and nitrate levels were also measured using Hawk AquaCheck water quality test strips. It should be noted that when conducting the chemical parameter test, all appropriate procedures were taken and precautions were also taken, as well as the proper disposing of all reagents. Zooplankton samples were collected by lowering an 80 micromesh plankton net down to the 1% light level. This net was then risen up through a water column, and the process was repeated 3 times to enhance the sample. Zooplankton samples were then observed and analyzed in the lab by taking 250 milliliters out of the collecting jar and placed under a Ward zooplankton counting wheel, which is then placed under a dissecting microscope. Zooplankton species would then be counted and identified through their appropriate species and down to the genus level where possible. It should be noted that as this project is a field study, there are no controls discussed in my results as there would be a typical lab experiment. The independent variable for this project would be the regions of the lake, while the dependent variable would be the physical, chemical, and biological parameters. Variables were attempted to be reduced by taking all the collections within a 2-week period in June, as well as it being within a 2-hour time frame of each other to reduce the variables that would cause fluctuations in the results. To reduce possible errors in measurement of my experiment, all the physical, chemical, and biological data was taken multiple times and then averaged. The average site depth was around 2.25 meters throughout the project, while the northern and central region had 1% light level around 2 meters, and the southern region being around 1.5 meters. The decrease in light level in the southern region would most likely be due to the murkier waters caused by runoff in the nearby area. The average temperature showed a general increase. The average air temperature showed a general increase from the northern region to the southern region, while the average water temperature showed a decrease from the northern region to the southern region. The increase in air temperature could best be explained by the 2-hour time frame, which would allow for the temperature to gradually increase, while the decrease from the northern region to the central region could best be explained by the presence of CWLP in the northern region, which emits warmer waters, which would cause the waters nearby the CWLP to be warmer, and the areas, as the water gets further away from that area, it would cool off. The average impinging light levels showed little fluctuations throughout the test sites and can best be explained by the 2-hour time period, while the average pH levels showed minimal fluctuations as well, with the maximum difference being 0.3. The average carbon dioxide levels were highest in the northern region and lowest in the southern region, and it rose up a bit towards the lowest in the central region, then rose up a bit towards the southern region, while the dissolved oxygen showed an opposite, with a lower number being in the northern region, a higher one in the central region, then lower in the southern again. This could best be explained by the metabolic reactions of photosynthesis and cellular respiration occurring within the aquatic organisms in the lake. In general, increased photosynthesis rates will increase the amount of oxygen while decreasing the amount of carbon dioxide, while increased cellular respiration will have the exact opposite effect. Increase the amount of carbon dioxide will decrease the amount of oxygen. Both nitrate and phosphate levels showed a general decrease from the northern region to the southern region, and this can be best explained again by the presence of CWLP in the northern region, as research has proven that the emissions from fossil fuel burning plants as well as water purification plants can contribute to a abundance in these nutrients. As predicted in the original hypothesis, the various zooplankton populations did experience certain regional variations. Both qualitative and quantitative variations were seen. In order to quantify the zooplankton data, the total tally of each group was plugged into this formula seen here. With the exception of rotifers, the average zooplankton population size is decreased from the northern region to the southern region. This pattern can perhaps be explained by the similar pattern in the phosphate and nitrate levels, as they also showed a general decrease from the northern to southern region. Concerning the qualitative aspect of the zooplankton data, both adult copepods and Cladoceran exhibited regional variations among the genera observed, while there was no regional variation among rotifers, as only one genera was really identified, and also the copepodnopoli were not classified into genera. Only two genera of Cladocera, that being Bosmina and Daphnia, were identified in both the north and central region, while the two additional genera, that being Leptodora and Diaphanosoma, were also observed in the southern region. Bosmina made up the largest number in all three regions, while Daphnia had the second largest number in both the north and central regions. One possible explanation for this qualitative variation of Cladocera is that both Bosmina and Daphnia have been proven to be seen in areas with higher phosphate and nitrate levels, and since the phosphate and nitrate levels were highest in the central region, that would best explain why their numbers were also highest in the central region and decreased as the phosphate and nitrate levels got less and less in the central and southern region. Only two genera of adult copepods, that being Cyclops and Dioptimus, were observed, and only one genera of rotifers, that being Caratel, was observed. These populations showed to be seen in all three of the regions, and the variations of these is not really best explained by what we've seen in the project, and in order to kind of explain what's happening with these populations, there would need to be a study of phytoplankton species, which most zooplankton feed upon, and that would best explain what would be happening with these and what would be resulting in the fluctuations. So in summary, my original hypothesis that the various zooplankton populations in Lake Springfield should exhibit certain regional variations was in fact confirmed and supported by the data in this project. It is obvious that additional research and experimentation of zooplankton populations in Lake Springfield is needed to better understand what is going on with these populations and why and how it affects the lake's health and the ecosystem's health. Additional studies of phytoplankton populations would also benefit the explanation of what is happening with these zooplankton populations and just all the zooplankton populations among the lake. A zooplankton study for next year's project would be the inclusion of a process called psychomorphosis, which is a predator rewarding process that the Daphnia go through, and I want to kind of see if this process exists in Lake Springfield, and if it does, over what season is it most abundant in. And then the next year I plan to look at the existence of vertical migration of zooplankton populations in Lake Springfield, which basically just shows how they migrate vertically throughout the day. But this concludes our presentation. Thank you for your attention.